I love me some histology. We're going to talk about peripheral nerves. Nerve fibers are essentially tracks, similar to the roads or the freeways on the highways. They're going to various places, some short, some long distance. And we have two locations, the central nervous system, where the axons are sheathed by special glial cells, which facilitate axonal function. And the myelin that's produced in the central nervous system is called the oligodendrocytes, which we will talk about in a later lecture. In the peripheral nervous system, axons are sheathed by Schwann cells. So again, please know the difference, oligodendrocytes versus Schwann cells that produce myelin. When it comes to Schwann cells, they're derived from the neural crest. Of course, they're located in the peripheral nervous system. Their main role is to form the myelin sheath, which causes a covering and protective effect, as well as conduction for nerve impulses. They support myelinated as well as unmyelinated nerve fibers, basically the axons. The image that I show you on the right-hand side here shows you a cross-section of the myelin. Sometimes you can see a layering effect. And then you can also see here this indentation. This is a longitudinal cut where you see indentations of the myelin. And those are called nodes of Ranvier, which we'll discuss later on. During myelination, the axon lies in a groove on the Schwann cell surface, and this is called the periaxonal plasma membrane. When the axon is completely enclosed by the cell, this develops a mesoaxon or mesaxon. Mesaxon membrane eventually wraps around the axons in a layering effect. Eventually, they will be compacted myelin layers as the layers develop, and then you can have up to 100 layers in this region, which serves as insulation as well as conduction of nerve impulses. Do note that the Schwann cell nucleus and cytoplasm are located in the outermost layer. Next, we're going to look at the ultrastructure of the Schwann cell. On transmission electron microscopy, we see a large axon that is wrapped by multiple layers of myelin. So basically what you're seeing here, that's the axon, and then you have numerous layers, which causes the thickening effect of that myelin layer. Newly formed myelin, which is also here, you can see that here's a Schwann cell on the outer rim, the nucleus is on the outer rim, and you see a thinner layer. So eventually it's going to layer up a lot more and thicken to become this more lamellated form of myelination. This inset here shows a higher power, higher magnification view of the neurofilaments and the microtubules that are present within the axon. This is actually, remember, this is the neuron, right? So this is the Schwann cell neuron here. This is the Schwann cell cytoplasm, and just imagine it's just wrapping the myelin sheath around that axon. So you also have another axon here, also developing into another thicker myelin sheath. So this is very early. So this is baby bear, this is mama bear, and this is papa bear right here. The unmyelinated axon that you can see over here, UM, has the Schwann cell on the outer side. This is a smaller caliber axon here. And so usually, don't forget, unmyelinated is kind of like a misnomer. Basically, the Schwann cells can enclose around the axon, forming a protective barrier, right? And it's usually just one cell layer or one layer of myelin. So definitely unmyelinated in the peripheral nervous system does have a coating. Now, in the CNS, right, unmyelinated axons are truly unmyelinated. Next, we're going to talk about the nodes of Ranvier, or also called the nodal gaps. Myelination is not contiguous along the length of the axon. You have nodes of Ranvier. Here's, again, one area here, node of Ranvier, where these are small spaces between each myelin unit. So this layering of the myelin on one segment here, layering of myelin on the other segment here, so there is a gap that occurs there. So like I mentioned before, in the CNS, the unmyelination areas are truly bare, whereas in the PNS, they're not really bare. Okay, so you do have some type of interdigitating aspect of the extensions of the Schwann cell cytoplasm, kind of like interlocking your fingers together at these nodes of Ranvier. So even though there's a little bit of a dipit here, there's still some cell structure that is still coating that axonal fiber. And so again, there is on the top surface here, the basal lamina is also called a basement membrane. So you still have a coating of the continuous basal lamina that goes from one layer across the node of Ranvier to the other layer as well too. So again, there is a little dipit there, which means that is a segment of the node of Ranvier. There's just not a lot of myelination there to be present. With nodes of Ranvier, you can have a saltatory conduction where nerve impulses have rapid movement from node to node. And these are regulated by voltage-gated sodium channels, which cause impulse conduction. There is a renewal of action potential, which gives rise to faster conduction. You can see how myelin is covering the axonal fibers. Don't forget that certain cells like the oligodendrocytes and the Schwann cells are responsible for myelin formation. So let's now add some clinical correlations. In this course, I don't need you to know the course conditions in detail, but do apply it. What happens when we lose that myelination process? So we have certain demyelinating conditions, such as multiple sclerosis and Guillain-Barré syndrome. So there is some type of myelin disruption. So when we lack myelin, remember, that's not only insulation, but it helps with axonal transmission and conduction. So what results here is slowed conduction down the axon. And so with multiple sclerosis, there's a demyelination in the CNS. It's an autoimmune condition that attacks the oligodendrocytes, and patients usually presents with neurological, sensory, and motor deficits. So keep in mind that in the CNS, right, neurons are unable to communicate with each other, right? And we talked about the axonal transmissions, axonal dendritic transmissions in the neurons lecture, right? So just imagine those conductions are slowed, and that's going to cause various neurologic deficits. In Guillain-Barré syndrome, it's a demyelination in the PNS affecting the Schwann cells. So these patients can present with muscle weakness. And why are the muscles weak? We don't have control of the muscles anymore because the neurons are not transmitting their impulses down the axons in a proper speed. Next, how are nerve fibers arranged in the PNS, right? Nerve fibers or axonal nerve fibers are grouped in bundles to create your nerves. So whenever you're talking in anatomy lab and you're talking about the vagus nerve, the radial nerve, this is what you're seeing, right? So we're going to look at the different layers and structures going from outside to in. The outermost covering of the nerve is going to be your epineurum. If you ever see this word epineurum, epi means outermost, like epidermis, right? So epineurum is this kind of gray, tannish gray outer covering here. Now, as we go further in, right, we have groups of axonal fibers arranged in fascicles. And so covering that fascicle is going to be a connective tissue called perineurium. So peri basically means around or surrounding. So perineurium surrounds the fascicles. Now, this is a magnifying glass, and going further in, looking at these little Cheerio or donut-shaped structures here, pathologists and food. So those are individual axons with their covering of the Schwann cells and the endoneurium. So endo means further in. Endo means further in. So each axon is covered by an endoneurium. So just to recap, we have epineurium going from out to in, your perineurium, which covers your fascicles, and then within the fascicles are numerous axons that are covered by endoneurium. Discuss in more detail about the epineurium, there are actually two layers. Epineurium is a very dense connective tissue containing collagen and scattered fibroblasts. We'll talk more about collagen when we get into the collagen section in the next few weeks. However, you can see here that the epineurium, it's kind of like a fascia in a way, but basically it coats the outer grouping of these numerous fascicles. And so when you're looking at the nerve next time in your anatomy, right, the covering of that nerve is going to be your epineurium. So again, it groups numerous nerve fascicles together in a bundle. Do know that there are two layers. The outermost layer is called the external or epifascicular. Remember we said that in the previous slide that epi means outer, right? So epifascicular means it's surrounding all the fascicles, right? The numerous fascicles here, and it's the outermost covering. But you can also see that there is an inner connective tissue that surrounds all these individual fascicle groups here, and that is the inner part of the epineurium called the internal or interfascicular epineurium. So it is also connective tissue, but it basically surrounds all the individual fascicles and their surrounding perineurium. Thus, in summary, be mindful of the organization. Now we're going to go from inside to out. We went from outside to in, and now we're going to go from inside to out just to kind of hash it out one more time. Each individual axon, right, contains your covering of your Schwann cells and your endoneurium, right? So an axon has the myelin sheath, Schwann cells, and the endoneurium. And so numerous numbers of axons are grouped together in a fascicle. So you see numerous little fascicles that are clumped together. So it's like just putting numerous pencils and crayons together and wrapping them with saran wrap. Right, so the fascicles are surrounded by the perineurium, right? Perineurium, and then the perineurium is the extension where you actually start to see blood vessels in the outer area. So it is considered the blood-nerve barrier because it is the barrier between the nervous tissue and the blood supply that is actually located in the internal aspect of the epineurium tissue. So on the outermost aspect, we have our epineurium. This outer layer is the external epineurium, the inner layer that contains the blood vessels and other connected, in the remainder of the internal connective tissue, right, covers the fascicles and the surrounding perineurium as well too. Now, just to look at histologic slides instead of figures, because I can give you an image, a histologic image to on the quizzes. So here we have two fascicles. And just know that within the fascicle, the bulk of the fascicle are numerous axons and their myelin Schwann cell sheaths, right? They are, the outer aspect here is going to be your epineurium, that red line there. It's going to cover the whole outer edge there. And then on the inner part, this looser connective tissue, that is your internal, and these are fascicles, just highlighting the rounded fascicles. And then on the internal connective tissue, you have your internal or your interfascicular epineurium. And then you can see, again, the perineurium is separating the fascicles from that internal epineurial tissue. Also, you may see some blood vessels surrounding there as well, too. This is a higher power magnification where you actually see the perineurium. On this side is probably the internal epineurium. And then you have numerous circles there, like I showed you those donuts or Cheerios in the previous slide, right? Those are the endoneurial outlines. And then within that center is you're going to have your axons and your myelin sheaths. So take a minute here to identify the structures. What is E, F, P, and what structure surrounds each axon and Schwann cells individually? Take a minute, and then we will discuss the answers in the next slide. So did you get it? Right? E is epineurium, which is the outermost layer. Fascicles are your bunches of your nerve fibers. Just think of the bunch of pencils. And then your perineurium surrounds the fascicles. And that's basically here, just to kind of outline that connective tissue right? That connective tissue that surrounds each bundle of fascicle. And then the endoneurium, you can't really see it on this one, you usually would see it microscopically, is basically the coating that surrounds the individual axon and Schwann cells. Yay! Woohoo! In this slide, I want to remind you not to have the lectures be viewed as individual silos. We want you to connect the dots, whether they are the lectures within the nervous tissue histology section, correlated with anatomy, physiology. And so, again, we talked about the neuron in the previous lecture, so this image shows you a slide of the different neurons. Eventually, we will talk about the neuromuscular junction and physiology. But from this lecture, I want you to be able to recognize Schwann cells, right? Oligodendrocytes, myelination process, and then the function of the myelin versus the nodes of RNBA, and even those connective tissue coverings that we talked about earlier. Lecture. On that note, contact me if you have any questions or need clarifications, and I will see you at the next lecture. Thank you. Bye!